Thank you, everyone. Yeah. Hi. So I'm really excited to be here today. Thanks so much for inviting me. It's always a great joy to come to these conferences because you do get to think more outside of the box. And so just to preface my talk a little bit, I'm actually trained in theoretical physics, so I think very much like a theoretical physicist would about life. And I'm interested in whether there are laws of life and what are the universal properties of life. And I take the assumption that life is actually an objective property of the physical world, so I don't think that me standing up here being alive is contingent on you observing me. It's not a subjective property. I actually am alive. And so I'm interested in how we can quantify that. So people don't really think we have like kind of a definitional approach to life. And what I'd like to see astrobiology do is to actually think about life more quantitatively and whether we actually can quantify life, how alive is a system, and then can we use that to actually quantify the origin of life transition or use that for more quantitative biosignatures, or even, as sort of the title of my talk alludes to, thinking about how we would reprogram other planets to be living worlds, which obviously gets into lots of ethical considerations and things that I think are very important, brought up earlier this morning, but I'm going to kind of focus on the science for this talk. So when I think about life, most people think about life as like a cellular phenomenon, and we're thinking about chemistry. But I don't think that life is specific to chemistry, right? So if you look at a planet of this, it's very clear which world is the living world, right? There's a way of thinking about life as a planetary scale process. Life has transformed the surface of our planet in a way that life has not transformed Jupiter, right? But both of those worlds have non-equilibrium dissipative structures on the surface. But in one case, it's cities, and in the other case, it's the Great Red Spot, right? So how do we think about what distinguishes those two worlds on that scale? And how can we think about it across scales? Is there some kind of way of thinking about what's happening inside cells and what's happening at a planetary scale in kind of a unified framework? So I am very much inspired by sort of a long lineage of physicists thinking about life from very, sort of quantitative or more conceptually and theoretically motivated perspectives. And so obviously Ernst Schrödinger is one of the most famous early proponents about thinking about life from this kind of deep conceptual foundations perspective. And a lot of people quote things from his book. He was obviously very inspirational in the discovery of DNA and other things with this book. But what really struck me about reading this book is his appeal at the end of the book to this idea of other laws of physics. And I think he was probably thinking specifically about the idea that non-equilibrium physics might need to be remodified to accommodate life. But I do think that we need to challenge our conceptions that our current conceptual frameworks, the way that we understand the world now, is adequate to describe life. And so what I'm going to do for the first part of my talk is kind of present some ideas that are meant to be provocative, because I think the thing is that we don't know how to challenge ourselves on this question, because it's very close to home. We are life. It's hard to think about ourselves. And it's also hard to think that we don't know what we are. So I'm going to kind of be a little bit philosophical for the first part of my talk, because I think it's really important to go down to the deep conceptual foundations. But then I'm going to come back up and talk about some of the quantitative tools that my group is doing to kind of think about life as a planetary scale process and thinking about life across all these scales and how we can unify that approach. So the first claim I'm going to make is that life is actually based on the concept of information. And so I think if we think about what information is, there's a lot of different ways that people talk about it. I'm not going to get into them in a short talk that I have today. But it is very clear that there is something very interesting about information in biology that doesn't happen in physics. And you might think about that as the origin of life transition itself, which is the way that we've been thinking about it a lot at ASU, is that you might actually quantify the origin of life somehow in a transition in the informational properties of matter. And I'm going to give a couple examples now of what I mean by information, which is probably not what everyone else means by information, but they're meant to get you thinking about the fact that life does something fundamentally different than other physical systems do. And my favorite example of that is, of course, us, because we're really fascinating physical systems. We do things that no other physical system does. And so I love the example that David Grinspoon has talked about quite a bit about this idea of anti-accretion, that we're literally the only planet we know that's making little metal boxes and launching them into space. Most planets accrete. A lot of planets don't anti-accrete. And if you think about the physics of that process, what's necessary for a planet to anti-accrete? It needs to have a technological civilization on the surface of that planet. I can't go back a slide now. Can you go back a slide? Sorry. And that technological civilization needs to have knowledge. It needs to have a very specific kind of knowledge. It needs to have knowledge about the regularities associated with gravitation. We call that knowledge the laws of physics, the laws of gravitation. But if you think about those are a compression of some kind of process that we understand in the world very well. Right? So there is this idea that this information actually acts in a catalyst in some sense for us to perform certain transformations. It's a catalyst for us to perform the transformation of launching satellites into space. That wouldn't happen without knowledge of the laws of gravitation. Just wouldn't happen. Another example is to think about the periodic table. A lot of people like to think about the periodic table in terms of the different physical processes in the universe that make different elements. Right? But there are some very heavy elements that are only produced by technology. This is why we talk about certain elements being actual technosignatures, right? We expect the only kind of physical process that could make them would be an intelligent physical process. So there are some things that the laws of physics and chemistry can't do alone that require physical entities that have information about those processes or some way of performing those sets of transformations, some kind of algorithm for actually doing that procedure. So I think this kind of distinction between these two kinds of physics is best captured by this quote from David Deutsch at University of Oxford. And he says, base metals can be transmuted into gold by stars and by intelligent beings who understand the processes that power stars and by nothing else in the universe. So if you think about that for a second, I think it's a really nice way of capturing this set of ideas because there are the objective laws of physics that we know so far that describe the world without observers. You know, we're the observers in the universe writing these laws down. And then by doing that process, we can actually do things. We can control certain processes, right? And this is what, when we're talking about looking for intelligent civilizations, we're talking about other physical systems like us that have knowledge about how the universe works and does the kind of things that we do as a technological civilization. And so I think those are really two different domains of physics. And I think we have a pretty good handle on the former, but not the latter. And there's a really interesting transition happening now as people are starting to think about consciousness and life from a more complex systems perspective, are thinking about these things kind of more rooted in quantitative approaches, that we actually might start to get at this latter kind of physics and really understand more about what we are, and also solve the origin of life, hopefully. So this gets back to the origin of life question. If you're thinking about the origin of life from this perspective, I've been using human examples because I think humans are a really interesting example of living physics. But the question is, how did all of those kind of processes emerge in the first place? And if you go back to the origin of life, there is a way of thinking about the origin of life itself as a transition in informational properties of matter, and that maybe information itself actually emerges with life. And when we talk about the laws of life, we're actually talking about the laws of information. And so I think analogies help to kind of illuminate this perspective. So people here are very comfortable because this idea has been around for hundreds of years about universal laws of gravitation, right? So that emerged from a human mind, right? Somebody had to think about that and create that. But we're comfortable with it now. So we have this idea of a universal law of gravitation. And when physicists today want to study that law or understand that property of nature, we study things like black holes. So we're all excited about the discovery this week. I think when we're thinking about life, life also has universal properties. There's a physics of information that's universal. But if you want to understand that, you go and study a living entity. So there's an analogy there about how would you actually think about the universal properties of life, the universal laws of life, and where would you actually go to study where that physics is most extreme or most evident. And so I think this gives us a set of tools for thinking about life from a really different perspective that can actually allow us to maybe open up the way we're thinking about things and dramatically change perhaps our approach to astrobiology and how we think about it. So most of what I want to talk about for the rest of my talk is actually this kind of approach to astrobiology that might get into some more quantitative things. And I make the argument that life is not physics as we know it because of these properties of information. Now, not everybody has to agree with that, but I think that taking that sort of perspective allows thinking in a different way. And I think thinking in a different way is really important in astrobiology, especially right now, as we're really on the precipice of the potential to discover life, but also as the point of this conference, on the precipice of trying to actually possibly spread our own life. We have to understand what it is. So I'm going to get into chemistry now, because chemistry is the scale of the physical world where life actually emerges. There's some interesting things with people thinking about life as chemical as a definition for life, but I think that's sort of confusing the concept of what scale does life emerge with what the definition of life is. And living systems obviously are chemical systems, but one question you might ask is, how are living systems different in the space of chemical transformations? Right? So I was talking about launching satellites into space as a transformation. You can also think about the chemistry in your cells as programmed transformations. The little script running the program is all the enzymes in your cells. Right? And so the metabolism that's happening right now for most people in this room after breakfast looks something like this. you've got all this chemistry happening. And we want to talk about how do we actually look for this chemistry on other worlds, or how do we do, like actually see this kind of chemistry on other worlds. And it might be a specific property of the molecules, but we always talk about this idea that life is an emergent property. It's really a property of the patterns of the molecules and the reactions, right? It's the two things together that make life. It's an emergent phenomena. So one question you might ask is, how do we actually study the emergent properties of these kind of chemical networks or chemical systems? And so being a physicist, I like to think about what are the simple models and what are the simple ways that we can think about things, and how do we generalize to these universal principles. And so one thing that has been really successful in physics is this idea of statistical physics. And so we are really familiar with the idea of an ideal gas in a box. And when we talk about those kind of approaches, we actually talk about ensembles, right? We talk about many systems and characterizing the statistical properties of those systems. Well, what if we could do that for chemistry? And the proper way of doing that in chemistry is not to think about the individual items themselves, but actually what transformations happen on them, what reactions are happening, what molecules are transforming into what other molecules, because that's what biology actually controls. And that is sort of what the living part of chemistry is. And so one thing you might ask is, if I put biochemistry in a box and non-biochemistry in a box, would I be able to tell the difference? So if I took an ensemble of all biochemical networks, all biochemical systems, and I had them, and I knew some statistical properties of them, would they be different than other chemical systems or not? We have some implicit assumptions that they would be, but we don't know by what measures. And so one of the things that we've been doing in my group is actually trying to think about the organization of life across different scales. And so we've gotten genomic data from about 22,000 different species, about 5,500 ecosystems, and actually planetary scale data cataloging all known enzymatically catalyzed reactions that are actually in the KEGG database. And we've constructed network representations, basically the patterns of the transformations and the reactions for all of those systems. And what we're looking for is whether there's any regularity there. So you can think of this as like my ensemble of ideal gases at a specific temperature, and the temperature characterizes the properties of that ensemble. I have an ensemble of biochemical networks, what characterizes properties. And so one of the ways that we're thinking about this is actually thinking about planetary scale organization of life. And if you think about that biosphere as being a set of chemical reactions, it's sort of like the hidden pattern of what biology is actually doing on our planet, right? This is a set of transformations biochemistry can do. There's a set of geochemical transformations, there's a set of atmospheric transformations, there's a set of industrial transformations, but this is biochemistry, right? So we can think about that as our biosphere. And then we can talk about, if it had universal properties, what would they look like? So there's several different hypotheses that you could use. And so the y-axis here is just some measure, like a network property or number of reactions, something that you can actually measure about the network or the chemistry. And then the x-axis is just the size of the biochemical system, so how many compounds it has. And the top row says it's a level-specific property. So if you look at how those properties vary with the size of the system, they might be different at every scale of biological organization and different from random chemistry. If you look at the middle row, it would suggest that it's a universal property of chemistry, so biology is not distinct from chemistry. And if you look at the bottom row, it suggests that biology is universal across scales and chemistry is somehow different. And so we actually did this analysis on the 28,000 networks or so that we had, and it turns out that biochemistry is very regular across scales. I won't get into the details, but you can see our paper that came out in Science Advances in January. And if you look at organisms and ecosystems, in various network properties, they look very regular. Ecosystems are very similar to individuals in terms of their network architecture, and the biosphere also looks very similar. If you compare to random chemistry, you actually have to do a random sampling of chemistry that samples with the frequency of reactions that we see across life on Earth to recover that scaling. So it seems to be that random networks don't have this property. It has to be very close to biology to have this property. So it does satisfy our criteria that biology is different than random chemical networks. And so what this suggests is that we can think about biochemistry in some kind of statistical sense and talk about all biochemistry on Earth being organized a certain way that's not characteristic of random chemistry. And so there's other properties that we've been looking at. I'm going to run out of time, so I'm going to kind of speed through this, but this is kind of work to come just as like a teaser. One is chirality, so you can look at just whether a molecule is chiral or achiral. There are scaling properties associated with that. It's very regular across all biochemistry as far as the patterns that biochemistry uses to transform chiral molecules into achiral or vice versa. And those patterns are very specific to the three domains. They use different kinds of patterns in their sets of reactions. We can also look at scaling of particular enzyme classes. And again, we see that biology uses very regular patterns in the number and ratio of enzymes for different reaction classes. So there does seem to be sort of a logic to the architecture of biochemical networks in this sense. Okay, so as promised in my title, what about planetary biochemistry on other worlds? So if we think these properties of biochemistry are actually universal, or capturing something about the physics of life, how information, in the sense of genetic information, has programmed biochemical reactions on Earth, can we do that on other worlds? And so I had a really clever grad student, Harrison Smith, that started this project when he was working with me about trying to think about putting biochemical networks from Earth in other environments. And so he started working with an undergrad, Alexa Drew, on thinking about Enceladus, because there's a pretty good catalog of what chemistry actually happens on Enceladus. And so basically what we did was we took biochemical networks from Earth, planted them in an environment that would be like Enceladus, and tried to figure out how much of biochemistry would actually be possible on Enceladus. Now, this is not accounting for kinetics or thermodynamics, but you just want to ask, is it impossible in principle for biochemical reactions to move through this reaction space in a different environment, another planetary environment? And it turns out the case is yes. If we take that biosphere, so say we could take all the enzymes on Earth and dump them in the Enceladus Ocean, what reactions would happen? Assuming all reactions were possible and we could engineer the enzymes in the right way to catalyze reactions under those conditions, we would get out over 4,000 complex biomolecules from the starting conditions on Enceladus. And that's probably not realistic because most of those reactions might not happen, but the idea of this is to start a framework for thinking about how to think about biochemistry on other planets and how we can actually think quantitatively about it. And so what we're trying to do with this is actually think about, if you take specific organisms, could they live? What compounds would you actually need to supplement them with if their biochemistry couldn't support it there? And in the future, what I really want to do is take the constraints from biochemistry on Earth and predict what other kind of chemistries could actually exist on other planets in a statistical sense. So we have the properties of what we expect for reactions and molecules to have a global topology, a structure like this on Earth. Now, if we start from Enceladus and we move through chemical space, not making any assumptions about what chemistry looks like, biochemistry on another world, could we actually predict what kinds of chemistries could live there? So there's different ways of thinking about panspermia from this perspective. And so it opens up sort of a new doorway for thinking about how we can actually program environments on other planets. And we did have some results on this, but I'm not going to talk about them in too much detail now because it's pretty preliminary and I'm running out of time, and I'd rather save time for a question if we have it. But I do want to thank my sponsors and also my lab group, who are amazing. And so Harrison's in this picture, also Hanju Kim that did the scaling work. And yeah, so I'll stop there. Thanks. Yeah, sure. I'll put this up here. Yes.
Hi, Bill Diamond from the SETI Institute.
Yeah.
Are you familiar with Jeremy England's work at
Yes.
MIT?
Yes.
Can you comment on that? Be interested in what you think about this notion of dissipation-driven adaptation.
Yeah, so I think it's probably a part of the story, but I'm always very deeply unsatisfied by non-equilibrium physics approaches to life because I think life is that plus something. It's clear that life takes advantage of a lot of physical processes. Like we have things like chemotaxis and gravitaxis, and humans use science. It's not like biology violates any known laws of physics, but I think they're not explanatory. I think you need some kind of understanding of algorithms and how information actually operates and transforms states of systems on top of that, but it has to be in a dissipative regime. So it might be part of the first steps, but it's insufficient. It would never explain, for example, you and I having this conversation right now. And I want that level of explanation for life. High standards.
Yeah, a fascinating discussion of the question of the difference of life from non-life in terms of information complexity. It leads me to sort of think of the ways in which living and non-living systems deal with entropy, of course, which is just the log of information. There's a lot of non-living systems that create low entropy processes. Obviously, snowflakes, for example, are very highly information complex. I was mulling over why it is that biology is different. Yeah. And for the most part, the non-living systems that generate low entropy products do so with thermal transformations. For example, you evaporate water thermally, then you freeze it, and you can create snowflakes. The difference of living systems is that they do this non-thermally. With this catalysis that you were talking about in so much detail, with many things. And I'm sort of mulling over non-living systems that do that, that use essentially photonic energy and using the low entropy of high energy photons to convert rather than the thermal energy. I could only think of 2 examples of that. One example is a very, very simple one. UV synthesis of ozone in the very upper atmosphere, and that's really the only biological system I can think of that does that. But possibly a more interesting example is the creation of tholins in the outer solar system, again presumably by UV photosynthesis. So this is the only other case I can think of is non-biological processes that are creating low entropy products without doing it in a thermal process. So it sort of makes me again think that the centrality of the understanding of tholins may be interesting in understanding how life and non-life are related.
Right. And I just one comment on that is that I think this like binary characterization of non-life life is a flawed approach to the problem, and that there's probably a gradation of states in between. So it might just be the tholene have what we would think of as like living physics, but it's a very, very simple example of it. So we wouldn't really want to call it life, but it's on the spectrum, right? So I think that's an important distinction to make as far as how we think about life.
Yeah, the real problem is the absence of examples that are in between life and non-life. There's almost nothing there. Viruses maybe, but of course viruses need life in order to replicate, so they're not really anything other than information transmission mechanism. So it would be nice if we could find more examples between. So thanks.
Last one over here.
I'd like to give a follow-up to the comment we just heard. There is a very important force acting in biology, which is called the entropic force. And this is the force that is due to the fact that if a hydrocarbon chain is forced into water, it organizes the water molecules around the chain and thereby reduces the entropy of the system. If the chains can get together, water and oil don't mix, we've all heard that, the entropy increases, and this is used by all life to stabilize the membranes that surround the cell and provide a barrier.
Yeah, so just a comment on entropy and like its application to life. So I think that like it's clear that life does these kind of things, there are the examples like this, but I think there's also a problem taking entropic concepts from physics and just like brute force applying them to life, because we count states a certain way in physics, and it might not be the right way of counting things once you get to more complex systems. And this is always a problem with information. Like when you talk about any kind of information measure, it depends on how you label things, right? And so if you want to count atoms, that's fine, but when you get to chemistry, you're ignoring the inner structure of the chemistry, and you need to start thinking about the transformations on the molecules themselves as countable properties. And I think this is why Lee Cronin's pathway assembly measure is really interesting as a complexity measure for biology, because it's actually trying to capture that inner structure and talk about quantifying actually the pathways through chemical space, not just the objects themselves. And I think that kind of approach is really important. So it's not necessarily the case that everything in physics doesn't apply to biology, but we do need to rethink the way we're applying those concepts to be broader and encompass kind of new approaches. Sure.
So I think that there was a very important point made that we don't see the systems that are complex but are not really life.
Yes.
And a very likely answer to this question is we never will on a planet that has life.
Right.
Because complex systems that are not life cannot, I mean, life will eat them. So it's more powerful, and we'll always eliminate them. Unless, you know, we can do it easily. You know, chemical engineers do it all the time, yes? So such systems exist.
Yeah.
And, but we will, you know, if we go to a planet that doesn't have life, we may see these kind of systems, and we may take them for life. So we have to be very careful in the way we interpret biosignatures.
Yeah, I agree with that. I actually think there probably are much more examples of cases around that are between non-life and life. We just don't know how to recognize them. And one of the problems, I think, is that we assume that an individual is a necessary criteria for life, but individuality itself is really hard to quantify. And our minds have been trained by evolution to recognize certain structure in the world, and we assign individuality to those structures, but they may not be really the relevant units of biology in some sense. So like all life might not be cellular, but it might require cellularity for a certain stage of complexity. And I think there's a lot of considerations there that are also really interesting. Thanks. Thank you.